So when it comes to diabetes, I told you earlier that the bottleneck is in the beta cell. And so we really need to learn more about what's going on in beta cells and what the nature of a dysfunction could be that would cause type 2 diabetes in beta cells. And in general, I like to look at it in three parts. Beta cells have to recognize nutrients like glucose and secrete insulin in response to them. So they need to sense the nutrient, they need to transmit a signal from the nutrient, and then they need to mobilize insulin granules. These are vesicles in the cells that have insulin inside of them, and they have to mobilize them to the plasma membrane in order to secrete the insulin. And so there's all kinds of cellular machinery involved in moving these granules to the membrane.
So what are the clinical implications of all this that I've told you about? First of all, that if we were to judge the reversibility of beta cell dysfunction by the intravenous glucose testing or by the estimates of beta cell mass that we have available, we would have to conclude that there is very little to be gained from beta cells in human type 2 diabetes because part of them are gone and the other ones are not responding. And I think this is the main concept that's being challenged. The reversibility of beta cell dysfunction. When we see deterioration of glucose control on a drug, we normally tend to write this off to the progression of the disease, to the natural history of the disease. This may simply be the accrual, the accumulation of glucose toxicity and insulin resistance, because in fact, we are now learning that much of the beta cell dysfunction can be corrected in patients with type 2 diabetes. And this lesson we've learned most evidently from bariatric surgery. This is, for example, a study of patients with long-standing type 2 diabetes on insulin who had failed on all treatments subjected to bariatric surgery. You see the blue shaded area is the response in non-diabetic individuals, and the red line is the beta cell glucose sensitivity before surgery, and then in green, 2 months and 1 year after surgery. You see the major improvement in beta cell glucose sensitivity as well as insulin sensitivity as measured by the clamp. So not only can we improve the insulin sensitivity, but either directly or indirectly through the improvement in insulin sensitivity, we can improve beta cell function. And the majority of these patients, this was not a large group, but the majority of them got off insulin, and they could be well controlled either on oral agents alone or with diet and exercise. So this is probably the extreme of a distribution which we are learning, that beta cell dysfunction can be corrected, possibly not to complete restoration depending on the duration of diabetes, but certainly to a clinically very useful extent.
Recently, there's been a new interest with the discovery of the KISS neuron. This produces kisspeptin, acting as a metabolic sensor sensitive to glucose levels, estrogen, leptin, and inflammation. And the KISS neuron sits upstream of the GnRH neuron, producing kisspeptin to increase GnRH, which in turn increases LHS and FHS levels, thereby stimulating testosterone production. This is a protein encoded by the KISS1 gene as a G-protein coupled receptor ligand for GPR54.
So we use this panel of metabolomics, metabolites, in samples of urine collections, 24-hour urines, from 2 groups of patients with diabetic kidney disease, which we divided up into a screening and validation group, and compared with a healthy control group, patients with diabetes but without kidney disease, and kidney disease patients without diabetes, to see if there is a signature that's consistent in patients with diabetic kidney disease. And of course, we had to correct for multiple testing. So this is the data that we found so far in terms of the screening group 17 of the 120 metabolites or so that we measured survived multiple testing in the screening group, and of those, 13 survived multiple testing in the validation group. So this group of metabolites we are now calling as a metabolomic signature of diabetic kidney disease. I don't think it is complete, as many more metabolites may likely be added to this panel as well, but at least it's a starting point of differentiating patients with diabetic kidney disease from healthy controls.
Let me say from the outset that the most difficult research you can do is clinical tropical medicine research. Your staff are at risk of the diseases they're researching. There are language difficulties, cultural difficulties, security difficulties, and even corruption, and we've been touched by all those when we've been working in these countries.
This anti-carbohydrates thing, the real message is eat more fat, especially saturated fat.
So the key question is, what effect does the carbohydrate content of the diet have on endogenous hepatic glucose production, which is the primary abnormality in diabetes? This is the paper you might want to look at, and this is the question. Is it increased hepatic glucose production or reduced muscle glucose uptake that cause hyperglycemia? And most of you believe it's this. You're told that it's peripheral insulin resistance that's causing the problem, and you've just forgotten the liver. The reality is, if this is the problem, you would never have hyperglycemia. Listen to that. If that was the problem, you'd never have hyperglycemia, because the homeostat would say just cut down the glucose production. So if you have hyperglycemia, you have to have excessive liver glucose production. It has to be there.
If somebody on a low-fibre diet needs to obtain 30 grams of dietary fibre per day, they'd have to eat at least 12 cups of green leafy vegetables. I know which one I'd prefer. And then there's the emerging evidence that the fibre that we eat is very important as a food source for the bacteria in our gut. The bacteria in our gut metabolise the fibre that we eat and produce a number of compounds that are very important to the health of people with diabetes. And there's an article there that I would recommend because it's quite good. But the bottom line is that people consuming low carbohydrate diets, we know they've got an altered gut bacteria. They have fewer beneficial microorganisms compared to those eating a high fibre diet.
The very low carbohydrate ketogenic diet delivered lower blood glucose, and it also demonstrated better improvements in HbA1c. It also had more body weight loss, the BMI was reduced, waist circumference was reduced, and there were also significant reductions in atherogenic dyslipidemia.
The funny thing is that when you look at these recommendations in greater depth, the answer is there. It's saying the amount of carbohydrate has a larger effect on glucose than the quality. The amount of carbohydrate has so therefore reduce the amount. But we miss that simple point.
The beta cell is an extremely complex, very sophisticated, highly differentiated cell. This is actually a painting by Kandinsky in 1938, and one wonders, how did he know about beta cells back then?
It was informative. I think it was very entertaining, and I would like you to join me in thanking everybody in participating.